Hello everyone, welcome back to MMBS111, Introductory Microbiology. This is mini lecture 6.2, and we're going to be talking about factors within the environment that can impact microbes. There are 5 learning objectives for this mini lecture. First, list and define 5 terms used to express a microbe's optimal growth temperature. Second, summarize 3 ways in which microorganisms function in the presence of differing oxygen conditions. 3, name 3 environmental factors besides oxygen and temperature that microbes must cope with. 4, describe the 5 associations present among organisms. And 5, discuss characteristics of biofilms. So first we're going to talk about temperature. Temperature is a big deal to microorganisms. So the cardinal temperature is the range of temperatures that a microorganism can grow in. And we have 3 different points in this cardinal temperature that sort of define what is happening as far as microbial growth. The first is the minimum temperature, and at minimum temperature, the membrane is kind of gelling. So transport processes are so slow that the microorganism isn't growing. Once that temperature begins to increase above the minimum temperature, then the enzymatic reactions that help to encourage microbial growth and metabolize nutrients, these reactions are starting to increase, and they're happening more frequently as the temperature rises. Once we reach the peak of this growth curve, this is optimum temperature once we reach the peak. And this is where enzymatic reactions are occurring at the maximal possible rate. They couldn't happen any faster. This is the optimum growth temperature for our cells. Once we reach maximum temperature, this is when proteins are going to begin to denature, the cytoplasmic membrane is going to collapse, and we have thermolysis. The cell is going to rupture. So our organisms are really looking for places within the environment at this optimum or just below optimum temperature range. Now, we can group microorganisms based on their temperature. These are called ecological groupings because these organisms like to live in areas of these certain temperature ranges. So at the low end of the temperature scale are the psychrophiles, and their optimum growth temperature is less than 15 degrees Celsius. So these organisms like it cold. Refrigerator temperatures generally incubate these organisms, so they tend to grow in the refrigerator. Most of these organisms, however, are not pathogenic. We typically find them in places like mountain lakes, rivers, snowfields, polar ice, and deep in the ocean, any areas where that temperature is going to be fairly cold. Then we have the psychrotrophs. These organisms prefer temperatures between 15 and 30 degrees Celsius. Staph aureus, Listeria monocytogenes, big organism in this group. They are known to grow in the refrigerator and cause foodborne disease. Listeria can even grow in the freezer. It is a common pathogen in things like uncooked hot dogs, ice cream. So then right in the middle of the temperature band, we have the mesophiles. And these are the majority of the medically significant microbes. Human pathogens have an optimal body temperature of 30 to 40 degrees Celsius. That's our normal body temperature. We find these organisms in plants, soil, water. There is a subgroup of mesophiles called the thermodurics, and these are organisms which are temporarily heat-resistant spore formers. So these are the organisms like the Clostridium group that can produce spores and are temporarily resistant to high heat. The next group are the thermophiles, and these are organisms that prefer temperatures just less than 45 degrees Celsius. They are, sorry, just above 45 degrees Celsius. Can't read the signs. They are found in soil and water, typically associated with things like volcanic activity, geothermal vents, compost piles, any habitats that are exposed to direct sun. And then finally we have the extreme thermophiles, and these are organisms whose optimal growth temperature is between 80 and 121 degrees Celsius. So incredibly high temperatures for these organisms. This is an image from your text that shows you where these ecological groups can be found and where their maximum and minimum temperature points are. You can see with the dots, the blue dots on the graph show you the minimum temperature, and then the red dots show you the maximum temperature. That's where denaturation is going to happen of cell proteins. And then remember, at the peak, this is the optimum. This is where the organisms like to be. All right, so let's talk about gases. Oxygen is a big factor when we're talking about microbes. And some microbes need oxygen in the environment, and others would really grow better without it. So the two primary gases in the atmosphere that are going to affect microbial growth are oxygen and carbon dioxide. Oxygen typically, like I said, has the greatest impact. And our organisms are going to fall into one of three categories. Either organisms that both use oxygen and can detoxify it, organisms that can do neither, they don't use it and they cannot detoxify it, and finally organisms that don't use it but are able to detoxify it. So we have five major groupings of organisms based on their ability to use and detoxify oxygen. The first group of organisms are the obligate aerobes, and they require oxygen in order to grow. If oxygen is not present, they're not able to grow. So if you're looking at growth within a broth tube, these organisms are going to grow near the top where the highest concentration of oxygen is going to be present. Our second group are the facultative anaerobes. These organisms can both grow with or without oxygen. However, they grow better in the presence of oxygen. So you're going to have growth, a large amount of growth near the top of the tube where the greatest amount of oxygen is, but you'll also have organisms growing throughout the test tube. Then we have the obligate anaerobes. These organisms cannot grow in the presence of oxygen. So in a tube, you're going to find growth at the bottom of the tube where there is no oxygen, but you won't find growth anywhere else. Our aerotolerant anaerobes, are anaerobic organisms. They prefer to grow without oxygen. However, they are able to grow in the presence of oxygen because while they don't use oxygen, they are able to detoxify it. So you're going to see organisms growing throughout the test tube evenly. Oxygen doesn't really impact their growth. And then finally, we have the microaerophiles. They need oxygen to grow, but in very low concentrations. So you're going to find them growing in the tube where oxygen is present, but in very low amounts. Okay, so one of the other factors that microorganisms have to address in their environment is pH. pH is the degree of acidity or alkalinity within a solution, and it is a measure of hydrogen ions within a liquid medium, within a solution. The pH scale ranges from 0 to 14, so low numbers show a acidic environment, while high numbers show a more alkaline environment. So when we have high acidity, we also have large amounts of hydrogen ions. When a solution is alkaline, we have low numbers of hydrogen ions. The majority of organisms prefer to grow in neutral habitats, anywhere from pH of 6 to pH of 8, in that range. There are some organism groups that like acidic environments. We call them the acidophiles. Many molds and yeasts are acid tolerant, which is why they are able to spoil foods that are more acidic, like any pickled foods. There are also a number of organisms that naturally live in your stomach that are acid tolerant. We also have alkalinophiles. These are organisms that prefer alkaline conditions. The Proteus species can create alkaline conditions in order to neutralize the acid in urine, and that is one of the ways that they're able to colonize the urinary tract and create UTI. Another environmental factor that microbes have to deal with is osmotic pressure. And osmotic pressure refers to the pressure that is exerted by solutions both inside and outside of the cell. This is the pressure that's put on the cell membrane. Ideally, a cell wants this pressure to be equal. We want equal pressure from the inside and the outside. However, if we change the environment, as we learned in mini lecture 6.1, if water flows in to the cell at a greater rate than it's flowing out, we're going to get a greater pressure on the inside than on the outside, and that can cause cells to rupture. We do have some groups of organisms that prefer to live in some unique osmotic pressure environments. These are the osmophiles, and they live in habitats where there is a high solute concentration in the environment, and generally that solute is salt. These are called the halophiles, and these organisms live in the ocean. They live in the Great Salt Lake. They live in environments where there is a high salt concentration in the environment. So some other environmental factors that our microbes have to deal with are radiation, light. Visible light can be damaging to microorganisms, just like it is to animals, and it can create burns and cause other problems. Visible light and other forms of radiation can be harmful to microorganisms. We have phototrophs, of course, that use that visible light as their energy source, but non-photosynthetic microbes aren't able to use that light and tend to be damaged by UV and other radiation. which is one way that we have learned to control microbial growth is through the use of ultraviolet light, which is able to kill many microbes and spores. Pressure is another thing that organisms have to deal with in the environment. And this has to do with like environmental atmospheric pressure. So we do have organisms that are able to survive and thrive in the deepest parts of the ocean, where atmospheric water pressure is incredibly high, 1,000 times the normal atmospheric pressure. So these organisms have adapted to live in these high pressure environments, but they generally die when exposed to normal atmospheric pressure. Then we have other organisms. So we have these non-living environmental factors that impact microbes, but then we also have other organisms that also impact microbes. In the majority of instances, microbes live in relationships with other organisms. Sometimes they live in a sort of symbiotic association with different microbes. Sometimes they live in association with multicellular organisms like plants and animals. These interactions between these organisms can be beneficial, or it can be harmful. Sometimes there is no effect at all. They just happily live together, but neither organism is harmed or benefited. And oftentimes these relationships are based on a nutritional interaction. One or both members needs some kind of nutrient from the other. So we have two general types of relationships among microbes, and those are symbiotic relationships, where the organisms live in very close nutritional relationships. One organism or both require the other in order to survive. Non-symbiotic relationships, the organisms are generally free living, and that association or relationship is not required for the survival of one or the other. There are three types of symbiotic relationships. Mutualism suggests that both members in the relationship benefit from the relationship. So both organisms are getting some kind of benefit from that relationship. Then we have a commensal relationship where one partner benefits, the other is not harmed by the relationship and doesn't necessarily benefit either. The human body is home to billions of commensal organisms that benefit from that relationship, but we aren't necessarily harmed by that relationship, and in many instances we don't really benefit from the relationship either. And then we have parasitism. Parasitic relationships, the organism that is the parasite is dependent on the other organism, its host, for its nutrition. It cannot survive without the host, and the host is generally harmed by that relationship. In our non-symbiotic relationships, we have two types, synergism and antagonism. In synergism, the members cooperate with each other, they share nutrients, they work together in this happy relationship. In antagonism, just as the term suggests, some members are inhibited, harmed, or destroyed even by other members of this relationship. And in some biofilms, that can be the case. So a biofilm is a mixed community of bacteria, and this is generally a synergistic relationship. The organisms attach to a surface and to one another, and there's this very happy cooperative relationship that occurs in a biofilm. The formation of a biofilm happens very simply with a pioneer bacteria that colonizes a surface. These are typically free-living, what we call planktonic organisms. They are free living, free to move around. But for some reason, they attach themselves to a surface. And once attached, these pioneer bacteria secrete an extracellular material, kind of like a slime layer, that helps to keep them attached to the surface. It also serves as an attachment point for other organisms to attach to as well. This creates what we call quorum sensing chemicals. Quorum sensing chemicals are released by these bacteria, and they kind of communicate to other organisms, hey, come join us, come colonize here. In most biofilms, you're going to have other species of organisms begin to attach and colonize within this extracellular matrix, and they will participate in quorum sensing as well with their own chemicals to attract other organisms. Biofilms are sort of a constant source of bacteria, so free organisms can be released from the biofilm, and then those free organisms are then able to go and find another surface to attach to and create a new biofilm. This is how plaques are formed in your teeth. This is also how organisms colonize transplant structures like heart valves, or if you have like a hip replacement, other structures that aren't necessarily part of you. And that's where we end mini lecture 6.2. Make sure you go through the questions here, that you can answer them correctly. Go back and check the learning objectives and go through those. Make sure that you are able to do each of those things, and hopefully this video was helpful.